Hi, today we're going to study mitosis. So we have worked the cell cycle. You have a general idea of the cell cycle and that it has phases G1, S, G2, which are all a part of interphase. And then it has a phase called M, followed by cytokinesis. So it's a phase called M that we're going to be working with today. And that phase called M is the division of the nucleus. So the division of the nucleus is really important because it's where all the genetic material is stored. And we know that that genetic material, that DNA, runs the cell. And so you have to have for each cell a copy of the DNA. And so it's really important for us to have one of each of the copies that were made in the S phase, excuse me, in the S phase of the cell cycle. One of those copies needs to make it to each of the new nuclei that are being formed in mitosis. And so we're going to go through the basic phases, and you don't have to have the names memorized, but you do have to have a general idea of them. It's not hard to memorize the names. You might as well while you're here, but basically you need to know what's going on. And so I just remember it, PMAT. And so PMAT is going to be where it's basically all parts of mitosis, and you're seeing what happens in each of these phases. So I'm just going to list them for you really quick, and then we'll talk about what happens in each of them. So prophase, metaphase, anaphase, and telophase. Okay. You might remember these if you're a Twilight fan from the first Twilight movie where they were checking out in a very angst-filled scene the phases of mitosis with the reward of winning a golden onion, whatever that was about. But anyway, so you are going to be identifying these, and we're going to be kind of going through in your lab about which one's fastest, which one the cell stays in longest, and all of that stuff. But for right now, we just need to talk about like what is happening in these phases. Okay, so we're going to start at the beginning. When we enter M phase, you're dealing with DNA that's already been copied. It's already been duplicated, so you have double the DNA material. Now, we don't consider it like twice as many chromosomes, but we do consider it like double the DNA material coming into this phase. So you've already got the setup, you're set up for it. But now we just need to split it. Okay, so in prophase, you're going to see your double DNA. So step one, if you don't want to memorize the names, although like I said, not too hard. In prophase, you're going to see your DNA, while it's been kind of like, kind of coiled, and there, it's a double helix, and you had, two of them, basically. Oh, that was bad. That were attached together somewhere at a centromere. If you haven't got to that yet, we will. We do that when we talk about the S phase. We talk about the centromere, and so you have this coming into it, but the DNA here, it's like too unwound. Like it's too loosey-goosey. And so what happens in prophase is you start to see it really coil up tightly, like coil up into a little ball. And so what happens then is your DNA actually becomes visible in the nucleus of the cells. Like you can see it through a microscope at this point. And so it would look like that. So see, they're attached together rather than like a big bowl of spaghetti. You've got condensing happening. And so your chromosomes then, or your sister chromatids, once again, if you haven't got to the S phase yet, we will. If you have, then you know. Your sister chromatids are condensing. So something weird is going to happen. For the first time ever, the nuclear membrane is going to disappear. And the DNA won't be inside of the nucleus for a very short amount of time. Now, it doesn't do that for long because that's dangerous. It's losing a layer of protection. But what happens is the nuclear membrane breaks completely down. So you start to lose your nuclear membrane. It's the membrane of the nucleus. So your nuclear membrane starts to basically decompose. Kind of a weird word. It basically breaks up. Okay, so your nuclear membrane starts to break up, and you'll see it kind of falling apart, and then you'll kind of see some spindle fibers forming. And your spindle fibers are kind of cool. Spindle forms. So your spindle forms, and you can't see that. Your spindle forms, which is going to be it's like the structural portion of the cell that helps these little chromatids to move to the right place. It's like the railroad tracks, if you will. Okay. So if you were to look at prophase under a microscope, it would look something like this. You would see, you could kind of see a nucleus, and you can kind of see some thick chromosome material in there. But it still looks pretty much like a normal cell because you haven't lost your nucleus completely, and the chromosomes are kind of just hanging out in the middle. So you can kind of see some daylight through, or some daylight, it's probably not daylight, some microscope light through where the genetic material used to be. So it gives that kind of spindly look. Okay. So after you got that happening, so now, well, that was prophase. Okay. So prophase, you kind of get this spindly look, ball in the middle there. Okay. And you're not going to be able to see, you're not going to be able to see the spindle fibers under a normal microscope, like a light microscope. They're just a little too hard to see. Okay, so then that's your prophase. Okay, and then next we would have then our PMAT. Remember PMAT? So next is metaphase. Okay, so in metaphase, now we start to see some organization. You start to see your chromosomes are going to line up in the middle. And they're lining up very organized. So I always think middle, meta, yeah, they're in the middle. And they're going to be very organized, and your spindle fibers are going to attach on either side. So you're going to start to see kind of like your little guys just line up here in the middle, wherever. I mean, they don't have to be like next to each other. And you're going to start to see spindle fibers attached to the centromere. Once again, you may not know what that is. Centromere is kind of a point in the chromosomes where they connect together as when they're in their sister chromatid form. Okay. And so you'll see that, and they're all nicely lined up in the middle, and they're ready to divide. Okay. And so this is what a metaphase would look like under a microscope. You can see that there's a nice line right down the middle. And it's pretty easy to tell. You can, like, the little spidery legs seem to be sticking out. Almost looks like little spider legs or octopus legs. Okay. So then that's metaphase. Anaphase. So this is actually more than the terms and the stages. I want you to get a progression. So it starts here in this little, where they all wind up tight. Then they line up in the middle. And then we start to see them. I shouldn't draw my little circle yet. We start to see them pull apart from each other. So they start to move apart from each other. And when they do that, you get this almost looks like a dragging motion, but studies are starting to show that it's not dragging as much as just movement. So your little leggies are so your two sister chromatids pull apart. Sister chromatids are what's made in replication in the S phase. So whenever you make a copy of the DNA, that copy of each chromosome is called the sister chromatid, and the original is called the other sister chromatid. So like two little sister chromatids that are attached together. And so sister chromatids move apart, and that's with the spindle. So you can kind of imagine, like, if you had them all like kind of leggy together in the middle, and then they started to pull, like if you pull on the center of it, you could kind of imagine that the little ends of the chromosome or the chromatids would like drag behind, so that you would kind of see it in that formation. So anaphase actually kind of looks like, I don't know, looks like jaws open under a microscope. Like here's a picture of anaphase under the microscope, and it looks like jaws that are kind of open, kind of like that, because that's the trailing chromosome little legs that you're seeing behind it. Okay. And so anaphase, they're apart, and that's how I remember anaphase. And then telophase is the final one. And so telophase, this is the final stage of mitosis, final phase of mitosis. Okay. Telophase is when the chromosomes are on either side of the cell now. The chromatids, they've split apart, and they are now in something, and in some sort of like you start to see two nuclei form. So you start to see the nuclear membrane come back, and then you kind of got the chromosomes kind of in the middle again, like a little ball like that. And that would be telophase. So basically you have them where they are going to be in their new nuclei. Now here's the hard part, is that this cell starts to pinch in. It's called cleavage, when it pinches in like that. And whenever it does that, right there, so it's going to break in half right there, that's called cytokinesis. Now, cytokinesis is not technically a phase of mitosis, okay? Cytokinesis is whenever you see like the cell actually, the cytoplasm actually pinching in half. The hard part is that it occurs at the same time as telophase. So you rarely ever can see one in telophase and one in cytokinesis because they're actually occurring simultaneously. So once you get into telophase, the cell splits like at the same time, and then the nuclear membrane reforms, and then there's two cells where there used to be one, okay? So that's basically how that occurs, okay? And so here's a picture of a cell under a microscope that's in telophase, and you can kind of see that it's got the two nuclei there, okay? And so then this is your phases of mitosis. Notice there's no interphase, that's before, and there's no cytokinesis, that's after. So this is where the M phase is, right there in between, okay? Interphase, remember, is G1S, G2, the cell cycle. Cytokinesis is the splitting of everything else, like the cytoplasm and all that stuff, okay? All right, so let's finish it off by talking about the numbers of chromosomes. This is something that's highly confusing. So if you have one cell, okay, if that cell has 6 chromosomes, obviously this is not a human. Humans have 42 chromosomes. That was a lie. Humans have 46 chromosomes. That was funny. I don't know why I said that. Humans have 46 chromosomes. This chromosome only has 6, okay? And so since we know that we have 2 copies of each, or most animals, most plants, most creatures, with exceptions, have 2 copies of each, then this would be like, this would have 3 different chromosomes that has one from mom and one from dad. So you have 2 number 1, one for mom and one from dad for number 2, 2 number 2, and one for mom and one from dad number 3, 2 number 3, for a grand total of 6. Okay, so we have something that's called our N number. Okay, so N equals the number of distinct chromosomes. So in humans, our N number is 23 because we have 23 distinct. What do I mean by distinct? Distinct means they code for different traits. So our chromosome number 1, think it's just a long strand of DNA, right? You know what DNA does. DNA makes mRNA, which makes proteins, which then drive everything around your cell, and including what you look like and how you're put together. And so the N number then would be the number of distinct chromosomes that are different from each other. So we have 1 chromosome 1 from dad and 1 chromosome 1 from mom. We call them paternal and maternal chromosomes. Okay, so we have 23 distinct chromosomes, with one exception. For boys, you have your 23rd pair is not really a pair, okay, because you have an X and a Y. And girls have X and an X. Okay, so then in what happens then is we go through the cell cycle, and so interphase occurs, and we go to the next stage. We still have 6 chromosomes. Now we call this 2N. Why do we call it 2N? Because it has 2 sets. That's like multiply. It has like 2 sets of distinct chromosomes, right? One paternal and one maternal. So that's why we call it 2N. Okay? When it goes through interphase, it still has 2N. Now remember, in the S phase, you've doubled the amount of DNA, but you still only have the 2 sets, like they're identical. And so they don't count for new chromosomes. You still have 2 you got from mom and 2 you got from dad. You didn't get like 2 new ones from some other random thing so that you have 4N. You have to be careful. That's what we have trouble with sometimes, is that people want to say, oh, well, when it goes through replication, then now you have 4N. No, you don't. You still have 2N. You still got the two number ones. One you got from mom, one you got from dad. There's just an identical copy of it. Okay, it's still attached together. It's nothing distinct or different. Okay, and so then when it goes through mitosis, then it splits. When they split and the cell splits in half and the chromosomes pull apart, now we call them their own distinct chromosomes. We now call them 2N for real. Okay, so they had to be pulled apart. Otherwise, they were actually part of one chromosome. They were like stuck together. Okay, and so then we call 2N is called a diploid cell, which just simply means two. It has 2 sets. 2 sets of chromosomes. Okay, and that is what happens in mitosis. And these are genetically identical. They should be. Sometimes mistakes happen, but we want them to be genetically identical. These are genetically identical daughter cells. So it forms 2 genetically identical daughter cells. Can you see that? No, of course not. Okay. So 2 genetically identical daughter cells is what's formed from a mitotic cell division. Okay. All right. So hopefully that's helpful. Just remember, you don't have to memorize the stages, but you have to know what happens in them. So I will ask you about them, but you can have a resource, like you can have something that helps you remember. I don't mind that at all. But then you do need to know what happens in each one. You need to understand the 2N and the N thing. And you need to be familiar with that, because when we talk about meiosis, it's different. And so you have to be able to tell the difference. Okay. I hope that helps. And if you have any questions, of course, as usual, let me know. And I will see you very soon. Have a great day.